we have a whole range of systems from the bench scale all the way to large scale. We have columns from bench to large scale. We have instruments from bench to large scale. I'm not going to go through all of them here with you. Our time is short and we want to be concise. But should you be interested, definitely reach out to us. And I'm sure for any scale that you're interested in, we have a solution. Okay, and so now we are going to get started with the topic at hand, which is, the advances in process chromatography systems. Now, why are we addressing this topic? Because for most people, when they are looking at a chromatography system, it's either a greenfield or brownfield site, or the equipment is older, and what do? They just want to replace it, right? And it's really the wrong choice to address the problem like that. So how do these engineering companies or MSAT or PD scientists choose this process chromatography automation equipment? Well, if they want to scale up, they just get a bigger piece of equipment, right, with higher flow rates. And they put a URS together and cut and paste from a URS that they had before. Worse, they take a vendor spec and convert it to a URS, and then ask their purchasing people to find out different vendors, get bids, prices, delivery, and they eliminate them based on some technology or previous experience they had, and they go ahead with it. Okay? Now, why do I say it's the wrong choice? Because the world as it existed 10 years ago isn't what it is today. The constraints that we face, cost, space, throughput, cycle times are very different, and your choice of equipment directly affects that. So if you look at this, this little plan I have down here, it's a facility blueprint, right? You have the upstream processing down at the bottom, turn the corner, and you have in blue the downstream suites. Then you have buffer making, column packing, etc., and two major corridors, right? Now, if the titers go up, guess what? DSP is stressed. We don't have enough space to fit in columns and process equipment. Maybe we can have a bigger column, sure. What about then you're going to need a bigger skid? What about buffers? You're going to need more buffers. So what do you see in these facilities? You have totes lined up, buffer totes lined up down this corridor, right? You're doing more cycles, bigger equipment, longer hours, more FTEs. So it's hardly a solution to what could potentially be a big problem. And of course, we are trying to reduce costs. We all say that, but what makes up the cost of making a biological drug? Well, of course, there's amortized costs of development, sure, and regulatory. But besides that, what's really under our control is the amortized capital cost, right? The facility cost, the equipment cost, consumables, and then, of course, most importantly, labor. Now, if you have a very complex workflow where you have 8 to 10 weeks per batch, right, like this on the left-hand side, different batch unit operations, and you have lots of hold points where you're getting QC done, and lots of manual intensive, whether it's column packing or buffer making operations, well, your labor costs are going to be huge. Your cycle times are going to be huge. So what can you do to address that? Well, your choice of equipment that you make definitely impacts that, and we'll see as we go forward. And this is a typical antibody process, manufacturing process, right? Going from cell culture centrifugation, chrome capture, viral inactivation, flow through steps, UFDF, formulation, fill, finish. You have hold points along the line, and you have buffer management, right, for all these different steps. So again, slow, laborious, traditional processes. And I've mentioned buffer making. When you look at any of these facilities, there's lots of detailed studies on it. If you look at the process down here in green, from cleaning the tanks of making buffers to getting the chemicals, checking out the chemicals, making the solutions, testing them, etc., storing them. All that goes into a traditional buffer making. That much operation is devoted to buffer making. Now you'll say, why this? I thought this was advances in chromatography. Why are we talking about buffers? Buffers are integral to making chromatography, to doing chromatography on a large scale, right? And here, for instance, there are 4 cell culture suites, 2 purification suites, 3 media and buffer prep, right? In these 2 facilities that Bioforum looked at. So about 30% of the operation is buffer making. And of course, all this, whether it's the capital costs, whether it's the FTEs for a particular plant, all go to make your cost of goods, right? Right down here. 8 weeks cycle time. And so you can make 8 batches a year, right? And even if there's overlap, you could do 10 batches a year. If you have a $150 million facility, that's 15 million contribution per year. If you have 70 people FTEs, that's 25 million contribution per year. You have materials and utilities, right? And the output of the plant is 100 kgs. That's $500 a gram. That's being very, very generous. It's usually 1,000 plus. So what can we do? Okay, what are the 7 critical advances that we want to address and talk about today? One is that when you get a chromatography system, you want the maximum range from those machines, right? So you can use smaller columns, medium columns, larger columns, as wide a scale as possible. That's not the case with most systems. I find people saying, okay, well, I know we are going to be running at 3 liters a minute. Let's get a 10 liter a minute machine. Well, doesn't work that way in case we have to scale up. Well, the problem is that machine then is not effective on the low end of the flow rate. So the pumps don't run very accurately when you're running at a liter a minute or 500 mils a minute or something like that. The turndown ratios are not there. So then you're running your process, which you're thinking you've designed for the future or equipment, but it's really being compromised because you're really running at a very low flow rate, right? So the repeatability isn't there. The variance is very high compared, because it's usually a function of the max output. And then what about safeguards? How many batches, if you look through batch failures, how many batches did you lose because you got air into the column or you had leaks in the lines? How much buffer do you really have to make? So, you kind of, and then how many people are there do you have just to oversee the run, even though it's an automated system, because of these kinds of issues, right? That's FTEs that you're using towards it. And then how about single-use systems? Do you use single-use systems? And if you do, how about using multiple size flow paths so you can have various ranges? What about Chrome systems now? Now, basic chromatography system will automate the basic operations like equilibration, washing, elution, regen, etc., right? You can change based on various parameters of time or UV or slope or run gradients, etc. But this day and age, automation is more complicated because your processes are connected. You're not running on islands anymore. Of course, there's 21 CFR Part 11 compliance. So your software has to be in compliance with that. But more and more, there's ITCSV, there's computer system validation, right? There are electronic signatures. There's connectivity with your network and Active Directory. So if your machines have different levels of access where somebody can only make a run, other people can, for instance, just do metrology and calibrate sensors. Those permissions are there on the machine, sure. But then how about, can you log in through your network right into the machine and still have those privileges? Okay, that's called Active Directory. How about connecting to the DCS through OPC UA? How about connecting to other equipment, whether it's tanks or another Chrome skid or whatever, through Profinet? So these kinds of advanced features are very, very important, and connectivity is more and more important as you make choices for equipment. And so that's something we'll discuss. And then, of course, process chromatography with inline stock buffer blending. We already addressed some of that, how much FTEs and space are required for buffer blending. So, what can chromatography do about that? How about continuous chromatography? We all know that baking one cookie at a time is not very efficient. Whereas if you have a whole line, continuous line, that's how things in most industries are made, and the cost can come down. Well, how about continuous processing and continuous chromatography? It's being talked about, and it's here and now, coming up to reduce the cost and bring more process control, more of the control into the process. And then customization. Everyone's processes are not the same. Right? Somebody, for instance, may want buffers added to their feedstock before they load onto the column. Well, they may do it in tanks outside. You may want to do it inline, right? You could have a program and develop and have your software so that skid does some of that, or UFDF, or multiple unit operations in one skid. So these are critical advances that exist today that I wanted you all to know about. So as you make your choices, you can make powerful choices that really impact the quality of your process and your cost and the complexity, and reduce the complexity, reduce the space, reduce the FTEs, and reduce time. So we have a whole range. I'm not going to go into all this, but we have a whole range of machines, chromatography systems, all the way from bench to very large scales, the largest of scales in the biotech industry. And small scale, 0 to as little as 100 mils a minute, 0 to 100 mils a minute. Okay, and then similarly, we have single-use systems going from very tiny scales, 0 to 100 mils a minute, with disposable sterile flow paths that you could use for gene therapy, for instance, all the way to much larger scales doing 1,000 liters an hour, again, with multiple flow paths. So again, I'm not going to go into details here. I'm just giving you an overview. So if you're interested, you're at least aware of what's out there in the universe. And if you choose to, you can engage with us. We'd be happy to discuss further. And then continuous chromatography. We have bench scale systems, large scale systems, buffer dilution systems, buffer blending systems, column packing systems, explosion-proof systems for people using organic solvents. So just to give you an example of the level of flexibility, and when I was talking about wide range, for instance, I'll take the example of multi-use systems are called trusty. So I'll take the example of our Quantisep Trusty 1000LX. You'd say, what is that? Well, it's a bench scale chromatography system which has 2 flow paths in one. What that means is that you have a 0 to 100 mil gradient system, so 2 pumps running 0 to 100 mils a minute each. And so the machine and on the software, you can choose whether you're going to run in the 0 to 100 mil a minute mode, or so it's a double-decker system. And on the bottom, you have 2 pumps, each running at 1 liter a minute. So you have, 8 inlets, 6 inlets on each, on each of the flow paths. You have 10 fractions on each of the flow paths and up to 3 devices, column devices, and sample injection loops. So essentially, you can choose with this machine, you can choose to do PD work and use small columns and run between 0 and 100 mils a minute. Or you could put bigger columns in here and run up to a liter a minute, fully sanitary, tri-clamp connections, the whole bit, and run at up to a liter a minute, right? With gradients. Okay, so full linear gradients. So it's effectively doing the job of these in other cases where somebody is using these two machines from one of our competitors, right? You would use this machine for a PD system going up to 600 mils a minute, and this machine out here going 0 to 150 mils a minute. If you're using both all the time, great. Then this is a good solution for you. If you're not, and you use this larger machine once in a while, then it's a wastage of space, wastage of money, wastage of FTEs, because on one machine with the same HMI, you can choose whichever flow path you want to run and do development work as well as scale up work in the same machine, right? That's what I'm talking about, is having this additional capability and having you be aware of what's really out there. And this shows you an inside look of the two small pumps and the two larger pumps at the bottom, the small flow paths with smaller valves and the larger valves, all with the same software, so you can choose whichever flow path you want to use. And then advanced capabilities, right? Everyone can make a method, do equilibration, wash, elution, sample, inject, fraction collection, have a fraction collector, collect a bunch of fractions, all that, right? You can automate, you can do things manually by clicking on the picture. Well, our systems would do that, but here's the deal, that if you're doing larger runs, what about bubbles? Air bubbles, you know that if you have a system running at a liter a minute, and air gets in, well, you can have an air sensor and that'll give you an alarm, it'll pause. Somebody has to do something about it. If you put a bubble trap, then the bubble trap has cleaning issues. You have to clean it. It has dilution issues. You certainly can't run gradients through the bubble trap. So that's a problem. And what we have is an auto air eject system, meaning that our system is smart. So it'll detect the beginning and the air bubble interface, the beginning and the end of the air bubble, and it ejects it out in real time. Okay? Thereby, air doesn't enter the column, but it's protected, and you can go ahead and make your run, and you don't have the issues of bubble trap. These systems also have the capabilities of inline dilution, right? So here's, for instance, inline dilution. They also have the ability to have pre-column filters, right? Depending on how your machine is configured, we have lots of custom configurations and options in the machines, right? So you can have inline pre-filtration. You can also have dual filters where if you're doing long runs where one gets clogged, it flips to the other and then back, right? And then inline dilution means you can have some kind of concentrated equilibration, wash, elution buffers and Wi-Fi along with your sample or harvest on one line. And after the harvest is loaded, then you can dilute in line and be able to run your diluted buffers, right? All our machines also come with leak sensors. If you're doing long overnight runs, these are fluid handling systems. You're running maybe development work at 100 mils a minute, or you're doing multiple cycles. You're running at a liter a minute. If any tubing leaks or pump seals fail or something like that, then you have an issue. Whereas our system, if there's more than 3 ml of liquid, depending on the size, 5 ml of liquid, that were to leak, the system would pause, give you an alarm, and pause. Right? So these are all very, very important advances in standard chromatography systems that you can avail of today. And then we have a whole range of single-use systems. Okay, and these are some very small systems. For instance, on the right here, you have an Adept. On the left here, you have an Adept 300, and that has two flow paths, one between 0 and 100 mils a minute, which is a 1.6 millimeter flow path, and then also a 3.2 millimeter flow path, right? So you can simply change this whole entire flow path out in about 15 minutes. And that's it. It's one contiguous flow path. And then you have larger systems, and then you have a 3 liter a minute system where you have 3 flow paths. So running between 0 and 100 mils a minute, 80 to 300 mils a minute, 30 to 300 mils a minute, 130 to 3 liters a minute, right? In the same machine. And you can have the ability in these machines also to do inline buffer dilution. And then these machines have been scaled up to over 1,000 liters per hour. Single use, single flow path. We've got videos on this. I'll just show you a small clip. I don't know if you can hear this or not. Well, I think we're going to pass. Yeah, so anyways, you just got a little bit of that movie. You can catch these videos live on YouTube. We're happy to send you links for these, but you get the point that they're very flexible and versatile machines. And the next advanced capability of these systems is the advanced software in process chromatography systems. Well, see, standard capabilities, you have standard capabilities that other vendors have, like operation from this graphical screen, right? You can click on the valves and pumps, enter the flow rates, and run manually, right? And then the flow path will light up. Or you can make recipes. Our recipes happen to look like little Excel, like a table, like an Excel spreadsheet, where each row is a chromatography step, and in the columns, in the individual cells, you can write down the valve you're flowing from, or the fraction you're going to, or the mode of change of the particular step, whether it's on time or volume or UV or pH, bed volumes, things like that. And you can see real-time chromatograms, linear and step gradients, alarms, right? Going to manual intervention, auto air ejection, inline calibration, HETP analysis, asymmetric. So those are all very standard features that everyone has. We have them too. But really the important ones and the more advanced ones are becoming more and more important are the fact that you have 21 CFR Part 11 compliance. So data is not erased, and it's around data integrity. And ITCSV, so computer system validation, electronic signatures, Active Directory, so you can log into the machine through your network as opposed to having standalone islands. How about interfacing with a plant DCS system, right? Uploading the data. Certainly data into a historian, no problem, but even into a DCS so that you can make decisions on what to do with upstream or downstream equipment, or maybe even connecting directly to tanks that might be in a different room through Profinet, right? Being able to look at what the pH or conductivity or levels are in those tanks and then making choices on whether to pump or not, or, give a low-level alarm or start filling the tank, whatever that might be, having those advanced capabilities, again, are very, very important. And I would encourage you to talk with your vendor before just sending out a URS and making your next purchase because the field has advanced. And then two very important things. Many people are starting to offer their chromatography systems with inline buffer dilution capability. Well, that's very good. So what that means is that instead of making a 1x equilibration buffer, you can make it, but at 5 times, let's say, the concentration, and then use the gradient feature in the machine to dilute that buffer. And sometimes, if your pH might be off because that happens in dilution, you can have a third pump with an acid or base so that you can get the pH under control. So that's a traditional inline buffer dilution system. And you can have that capability in your chromatography systems. We have that in our systems, and in our trustees, for instance, we call that capability Premier, right? So you have inline dilution capability in each of the machines. We also have something called stock buffer blending, where you can have actual stock concentrate. So you don't make a concentrated equilibration or wash or elution buffer. You just have the base buffers, right? So a monobasic phosphate and a dibasic phosphate, 1 molar, and then a 3 molar salt and Wi-Fi. And now if you want to make a 0.05 molar equilibration buffer, which is 0.05 molar phosphate at whatever X7 pH, well, guess what? The pumps move in such a ratio so they deliver that at point of use, right? With the right conductivity. And then the wash buffer is made. So you don't actually make concentrated versions of any of the buffers. You don't have to make buffer making at all. Just have stock concentrates and the buffers get made in line. So here, for instance, is a very large system. This is the one that one of our customers wanted for, is one of the largest systems of its kind in the world. It's 7,500 liters per hour. Okay, and these guys at that scale, making buffers this was for COVID vaccine, would have been crazy, right? So would have been very arduous. So this system, for instance, comes with 5 pumps, right? So buffer concentrate pumps, Wi-Fi, harvest pumps, you can load, and then you can make buffers, equilibration, wash, elution buffers in line. And here, for instance, with 200 millimolar salt, you're making 7 millisiemens equilibration buffer and 24 millisiemens wash buffer. You can see how quickly that's made, about 30 seconds. And then same thing with the pH. Whatever pH you choose can again be fulfilled. So the wash buffer, the equilibration buffer might be at a certain pH. The wash buffer might be at the same or different pH and different conductivities. And the systems make that. And we are running, in this case, up to 7,500 liters per hour. Right, so you saw the little systems running at 0 to 100 mils a minute. These are the large ones, and we have everything in between. Again, we have a video showing details of this of the system. I'm not gonna I'm not gonna uh, have you see the whole system. I'm just gonna show you some clips of the video. So essentially, what you have here in this kind of machine is the capability to do both chromatography. So this is a trustee elite, right? So what you have in this is the capability to do both chromatography and buffer blending, which is the same as two machines from our competitor, which is one is a chromatography system, and then the other is a buffer blending system. Both these functions, so in a normal bioprocess manufacturing environment, somebody would use this machine down here to make the buffers in totes, then take those totes to the purification suite where you'd have a machine like that doing chromatography and connect those buffers to that machine to do chromatography. Now here are the stock concentrates. So in our machine, you would simply have these stock concentrates and Wi-Fi connected to this machine, and you would do both chromatography and the buffers would be made in line, right? So you eliminate the need for both these machines, and you can replace it by a machine much smaller in footprint and with dual capability. And so you, of course, save time, save space, and you simplify your process and reduce your FTEs. So these systems have been used on very large scale. These systems have been used in very large scales. And today we are doing what will be the largest insulin plant in Asia, about 2.5 tons outside of Singapore. And the ability, of course, to use our columns, which are the feature of a previous webinar, along with these buffer management systems really reduces the footprint significantly, right? So reduces the buffer tanks from 6,000 liters to 2,000 liters, and they can go the entire, they don't have, and even in the 6,000 liters, they were making several batches of buffers, which they don't have to. They validated that the buffers could last as much as a week. And we are running, and then we have automated column packing systems and they talk with each other. They can integrate with each other, right? And the system software can integrate with can talk to the buffer holding tanks as well. So overall, as we had said earlier, the significant savings in space, right? We also, I'll come back to this in a few minutes, but we also have the ability to customize different unit operations. And so, for instance, in a flow-through skid, we can take a flow-through anion exchange and a HIC column as well as a nanofilter and be able to integrate that into one skid. But I'll come back to that in a few minutes. The next real advance that we want to address right now is continuous processing. So Sepragen's got end-to-end, people want to do end-to-end continuous processing. Continuous processing has come upstream where people are using perfusion bioreactors along with ATF, and they've developed something like a capture chromatography system with multiple columns, sometimes called SMBs, where you're loading one column second one may go in, while the first column is going into overload, the second one is being loaded, and the third column, which is presumably already loaded or saturated, is going through the wash, elution, and regen cycle. And then these flip around in a merry-go-round so that you have continuous feed and you have product being eluted out. So we have small-scale versions of these machines, not so small actually. Again, single use with multiple flow paths running 0 to 100 mil a minute. We have one system that does that. And in the same machine, you can put a bigger flow path, 3.2 millimeter flow path, and you could be going up to 300 mils a minute, which would work with a 250 liter perfusion bioreactor. Okay, so pretty substantial, but the footprint you can see is very, very small, but you can get large quantities of material, right? If you're doing even two bioreactor volumes a day, that's 500 liters, and at 2 grams a liter, that's a kilogram of product a day, right? From a small continuous line with a machine like that. And then we have larger versions of these machines, 3 to 5 liters a minute and 10 plus liters a minute. And we have these in multi-use and we have these in single use. And what we've also done is integrated inline buffer making capability into these systems. So you can see these 4 pumps here that make the buffers from the stock concentrates. Now, what is the benefit of all this? Well, if you're running batch, you can see the cycle times are very long. Even in fed batch, the cycle times are reduced if you overlap these different steps. And that's with medium titer. But now if you're doing continuous processing, you can significantly shrink down your cycle time and you can increase your productivity as much as an order of magnitude. And we'll address this right now. So here you can see the relative facility of a batch processing plant. And here's the facility size for a continuous process. You can see the number of totes and all that are considerably reduced. The footprint is considerably reduced in the downstream side for a continuous process. And the downstream is where the bottlenecks are, by the way. So you can have a process size be reduced by as much as 10x footprint. And then people have done models. This is not our data. Other people have done models, but showing that the total cost, could come down from, in this case, 300 euros to 39 euros, right? So relatively. And this is effectively a 12,000 liter facility could have the same output as a 200 liter continuous processing facility. Again, not our model, other people's. I'm just sharing that that's the trend of the industry. And we are in keeping with that whole trend. You can see here is our Fusion Elite system, which is a continuous triple column system. And we have this with inline buffer blending. There's no sense. People are trying to move to continuous processing, but there's no sense in going to continuous processing if you have to make the buffers in batches, because you've got to feed the beast, right? You're running continuously, a continuous capture, and that means that the system is continuously going to require buffers. So if you're going to make buffers down in the buffer room and move the totes over, then you're going to be running a bucket brigade trying to keep up with the system. So really the answer is to have integrated continuous on-demand buffer making available, and what better place than to have this Chromskid itself do it, which is what we have done here. It's the first, we are the only ones in the world to have done that, and it gives you enormous leverage and capability in terms of the power and productivity that you can bring into the purification suite. That's, of course, we are keeping track of the advances. That's we talked about 7 advances, and that's the 7th advance breakthrough that we talked about here. And this gives you a closer look at the HMI, or the human machine interface, for a machine like this, where the orange is the harvest line, which is loading onto one column, going through the second column. And then the buffers that are made in line, the equilibration wash buffer. It's not like they're made separately and we have intermediate holding tanks. It's all done seamlessly. The operator simply just programs in the continuous run, calls the equilibration wash elution steps, writes down what the molarity is of the different buffers and the pH and conductivity, and the buffers are made inline automatically, seamlessly, really. Right? And if there's buffer that's outside the spec, that buffer is ejected until it comes within the tolerance band. You get a process tolerance alarm. And you can see that the accuracy and the responsiveness with which the buffers are made. So here's from a 200 millisiemens, we are down to 10 millisiemens of equilibration buffer, 45 millisiemens here. The pH in the case of the first buffer was 7.2. The case of the second buffer is 3.5. So again, this is a very, very fast process. So it's under a minute. And we can have these in multi-use, or in this case, you see a machine which is our Adept version, or the Fusion Elite version, where you have single-use flow paths as well. So in this case, where you have continuous processing, the Fusion Elite, we talked about the trusty Elite, right? Where you had the chromatography system with buffer blending. Here, you have a continuous 3-column process with buffer blending. So again, this is the equivalent of 2 machines. So whichever competitor in the world you go to, there are 2-3 companies that make continuous processing machines. That's what you would get. And then in order to manage the buffers continuously, you'd have to have a separate machine like that. You'd have to make the buffers and then bring them over to this other machine. And essentially, we have a footprint which is smaller than both of these machines. You don't have 2 FTEs here and 2 FTEs here. You could have one person running this whole machine, right? And its capability and throughput and all that are equal to both these machines. Okay, so very powerful in terms of the capability that you can have. And we talked about cost, we talked about footprint, and we talked about FTEs. So the space savings is about 30% of the space savings of a whole facility. There's another video that we have on YouTube. You're welcome to watch it. Just in the interest of time, I'm not gonna show a lot of it. So there's a lot of detail. You can see this on our website online. I'm just flipping through just to give you a sense of it. You're welcome to go out. So here is, I didn't want to share the whole video with you. I'm going to let you take your time and review it at your leisure. Just wanted you to know that the resource is available for you, okay? So here's an example of the effect on the facility. Now, this is a next generation continuous processing facility, okay? state of the art, where what they are proposing is that they make buffers in a buffer room with inline buffer making machines. And then you have totes over here. And they're proposing that you make 3 to 5x concentrated totes for equilibration wash solution buffers. And here's the processing line, right? You have upstream, and then you have the chrome skids, the chrome skids, the chrome skids. So capture, flow through, flow through. And so by using this system that you just saw, the Fusion Elite, you can get rid of all this. You can get rid of all this. Maybe you have some totes against this wall, about half these totes against these wall, which are the stock solutions, and that's it. So all the space, which is 30% of the space of a biomanufacturing plant, is gone. You don't need these buffer making machines, which are, so if you look at it, it's $2,000 a square foot for processing facility, right? And it could be several $1,000,000 of space. The FTEs to manage this is 4-5 FTEs to manage this. You got these totes, the cost of all these totes, and these are single use, maybe, or they have to be clean, right? So they're single use, very expensive, and also not environment friendly. And then, of course, the capital costs of making these buffers and buffer making machines. So all that is gone. And in this facility, we estimate this is going to be about $7 to $8 million, about 3-4 FTEs, and about $4-5 million worth of space savings. That directly impacts your cost of goods, your workflow, and your productivity of the plant. And so these are just not random things that we have done. We've actually done 2 commercial lines. So what you see here are 2 antibody lines. The first one is a 3-column capture. The next one is for protein A. The next one is an AEX flow through and then a CEX. So these are 2 independent lines meant to run at 2,000 liter perfusion scale, right? And so if these technologies are used, we model that the cost of goods can come down from $400-500 to $130 a gram. So significant reduction in cost of goods, significant reduction in complexity and space savings and FTEs. So how can we at Sepragen help you intensify and simplify your process? You're welcome to give us a call. We can sign CDAs and have a one-on-one chat with you. We do complete documentation packages, FATs, SATs, IOQs, and all that. We do a lot of custom design and engineering work here, and we have custom manufacturing capabilities, full material traceability, and all that. And happy to engage with you. We've got customers around the world. Our products are used on 4 continents around the world. A whole lot of standard machines, but a lot of customized capability as well. Customized capability, where told you I'd come back to it, where we've got multiple unit operations on a single chrome skid, where you may be doing HIC, AEX and HIC combined onto a one skid, where the material flows through an AEX column and may have salt added to it so it can bind onto an HIC column. Or in chromatography, if you're just running chromatography, the harvest may need to be conditioned and you may want buffer added to it, right? So that it can then go on to the column. So we made machines like that where you can do that. Or in case you're using organic solvents, you may want explosion proof. Again, we worked with people to enable that to happen. So that's all for now, folks. If there are any questions, I would be happy to talk with you, take any questions. On the other hand, if you'd like to send us a note and want to talk with us confidentially, and have a confidential discussion about your needs and what we can do for you, happy to engage as well. With that, we are towards the end of our hour. If there are no more questions, I'd really like to thank you for joining us today. I hope that this was useful to you. You can see tremendous cost savings in terms of space, time savings. You saw those two machines that competitors, for instance, you might have to use to accomplish the same tasks. Again, cutting down the space by over 50% in many cases. Very powerful. Happy to engage with you. Thank you for joining us today, and have a good rest of your day. Bye-bye.